Okay. All right. So today we're talking about the innate immune system. So this is, of course, the very first line of defense against infections. All the mediators of the innate immune system are fully functioning um, prior to an encounter with an antigen, which contrasts it with the adaptive immune system, of course. There is no change in the quality of the immune response or the magnitude upon repeat exposures of the same antigen. That again is fully on the adaptive side. This is going to be the same response every time it sees a particular pathogen. And it's phylogenetically the oldest part of the immune system. It can see redundancies through all animals and even plants. Um, uh, throughout the plant and animal world. So it can prevent, control, and eliminate infection. It prevents both through physical and chemical barriers. It will recognize products of damaged and dead host cells to eliminate and initiate tissue repair. And it will stimulate the adaptive immune response and then influence the nature of the adaptive immune response. So it's obviously an extraordinarily important part of the immune system because it really sets the stage. We know the adaptive immune system is more robust, but the innate immune system has to be working well for that to happen. That cytokine milieu that kind of gets created by the innate immune system is probably responsible for a lot of what we see in autoimmunity and allergy, where we have that initial incorrect cytokine profile as the adaptive immune system is starting to do its job. Okay, so for recognition, this is a really important part of this chapter. Um, it recognizes the molecular structure that is characteristic of microbial pathogens. So examples of microbial pathogen structures would include PAMPs, which are pathogen-associated molecular patterns. It's just patterns we see that we tend to not see on animals. They tend to just be on pathogens. Examples would be double-stranded RNA or unmethylated CpG DNA or lipopolysaccharide or LPS. These patterns are essential for the microbial survival. Um, but ultimately, the innate immune system can never distinguish between specific pathogens. It just relies on recognizing these characteristic structures that are redundant through many, many, many different pathogens. It can also recognize our endogenous molecules that are produced by damaged cells. So, an example would be DAMPs. These are damage associated molecular patterns. So, these are things that would not be there in a normal, healthy host, but once damage comes to a cell, it's recognized so the innate immune system can help clear it. So there are pattern recognition receptors that recognize these different motifs, these PAMPs, these DAMPs. They're encoded in germline DNA. There does not need to be any change or adaptation for them to be fully functional. And they are ubiquitously expressed everywhere. Neutrophils, macrophages, dendritic cells, most cells in the body will have some sort of pattern recognition receptor. So here are some examples of pattern recognition receptors that we're going to talk about in a bit more detail. And a nice demonstration or a reminder that they are really everywhere. So these are extracellular that have the pattern recognition part of the receptor on the outside, signaling on the inside. So an example would be TLR or lectins. And then similarly, you can actually have TLRs or other similar receptors on the surfaces endosomes within the cell, or some that are just in the cytosol, like NLRs or RLRs. Okay, here is a nice table of all the different things that we're actually now going to talk about, all of which are important to know to some extent. I'm going to try and highlight what I think is important. And remember, what I think is important is always based on what we have identified as a problem and as an important area of immunity typically. So let's just kind of go through these one by one. So TLR stands for toll-like receptors. They are made of a binding domain that is rich in leucine. It's just the way we characterize some domains. So basic scientists usually can just understand the characteristics of it because it's redundant through many different receptors. And then there's a toll IL-1 receptor or TER homology domain for signaling. So here we can see just that. We see this leucine-rich binding domain on the outside, and then we see this TER signaling domain here on the inside. These are all the different toll-like receptors. And I think there are some properties of these that are important to remember. So one, I think it's important to remember which are on the outside of the cell and which are found on endosomes of the cell. And if you can start to remember what, if you can start to remember what each toll-like receptor actually recognizes, that will help you place it either on the outside of the cell or the inside of the cell. So, sorry, my dog is jumping all over me. Okay, the inside of the cell, of course, will have access to things that will not be available on the outside of the cell. So if a virus or a bacteria is breaking down, it's going to release RNA and DNA that would not be available unless it's broken down. So those more intuitively would be found on the inside of the cell once the cell, like a neutrophil, has had time to process it. So these are 3, 7, 8, 9. So that's one that I think is, I would start committing to memory. And, probably I would start making the differentiation of which each of these uh, recognizes. I do think that is somewhat important, although overall for the boards, they're moving away from kind of very basic science facts. But some of these are related to immune deficiency, and those are going to be important. These ones on the outside are more related to the structure of different bacteria, like lipopeptides, peptidoglycan, LPS, flagellin. These are all things that are going to be outside of the bacteria and therefore recognized exterior to the cell. Okay. Here is, I think the next two, yeah, are very, very important. Um, so there is a lot of redundancy in the way that the toll-like receptors signal and signal to the nucleus where it allows expression of different inflammatory genes like cytokines and chemokines and different co-stimulatory molecules that will allow the adaptive immune system to work. And by and large, they all signal through something called MIDI88, except for one, and that's TLR3. TLR3 is MIDI88 independent. And the reason that is important is because there is a known immune deficiency associated with MIDI88. And if that's gone, you lose all of the signaling except for just a little bit, because there are two that can signal, one that necessarily has to signal independent of MIDI88. That's 3. 4 has the option of signaling either way. So when you have a MIDI88 deficiency, you're not 100% loss of your TLR, but you have lost most of it. Okay. There is another immune deficiency that is also associated with the signaling pathway. It's a NEMO deficiency. And this one I've just tried to modify to show you where NEMO is and where it acts. But for NEMO, I believe the only one that will signal independently of NEMO is TLR9. Still MIDI88 dependent, but NEMO independent. This I like too because I really think it shows nicely, starting with this TER domain, the whole signaling pathway, starting with Mighty 88, IRAC, through NEMO, and then to these transcription factors here. NF kappa B, one of the most important ones we'll ever learn about in immunology. It's actually one of the only ones that we talk about. Um, and it will allow transcription of these genes for cytokines that are very much associated with the innate immune system, like IL-6, TNF, IL-1, IL-12. Okay, I'm going to skip over that one. And here is a list of immune deficiencies that I've put together, um, all part of those associated with TLR in some way. So if you have a NEMO or I-kappa B deficiency, you have an ectodermal dysplasia, and then you are susceptible to pyogenic bacteria and mycobacteria. You can have an MIDI 88 deficiency, same. You have pyogenic bacteria and mycobacterial susceptibility. IRAC4, that was another one of those intermediaries that is associated with an immune deficiency and makes you predispositioned to pyogenic bacteria. And then there's an UNC93B or TLR3 deficiency, which leads to HSV encephalitis, and a TLR4 that leads to meningitis sepsis. So these are very important in the immune deficiency world. We think about them less. They're certainly more rare. I have actually, when I was in Kansas City, I took care of one family of siblings that had ectodermal dysplasia with immune deficiency, although did not have gene mutations in either of these. They probably had something novel. But one of the more important things, I think, from this chapter is kind of starting to understand the TLR and the immune deficiencies that are associated with it. Okay. Um, NLRs, so the next set of immune deficiency, I'm sorry, the next set of receptors are more important for other subspecialties because they're more associated with autoinflammatory diseases, but we do need to know these as well. So there are NOD-like receptors are called NLR. They also have that leucine-rich binding domain, and they have a notch for binding to each other, and then they have a signaling domain. So there are three subfamilies, card, pyrin, and burr. Card is important because there, of course, was an immune deficiency identified and published with it. A card9 mutation, it leads to susceptibility to fungal infections, and it was identified in one family. This is a while ago now, 2009. One specific NLR you've probably heard of because of its association with Crohn's disease and Blau syndrome. It's NOD2. It's located in the panicles, the small intestines, and it stimulates expression of defensins, and it binds to something called muriamyl dipeptide. There is another set of receptors called CDSs or cytosolic DNA sensors. They detect double-stranded DNA in the cytosol, and they will initiate type 1 interferon through a STING pathway. It's a stimulator of interferon genes. If there's a mutation in STING, this leads to autoinflammatory syndromes. There are RIG-like receptors or RLRs. These are also cytosolic sensors, but instead of DNA, they sense viral RNA. Produce type 1 interferons, and examples are RIG1 and MDA5. Okay. There are NLRPs that form inflammasomes. These are complexes that form in the cytosol in response to infection, formed of various enzymes. And they generate IL-1 and IL-18, specifically IL-1 beta. NLRP3 mutation is associated with hereditary fever syndromes, including familial Mediterranean fever, because these are potent fever inducers, these cytokines, and CAPS. cryopyrin-associated periodic syndromes. I know we don't see these much, and our institution room sees them more. That's not the same for all institutions, but these are things that are absolutely within reach for testing on our boards. We treat them through IL-1 antagonism appropriately because we're trying to shut down the inflammasomes from making IL-1. The IL-1 antagonists, of course, are things like anakinra. Here is just an example of an inflammasome that is causing the generation of this inflammation that if it is if there is actually a DNA mutation and this is running unchecked, then people can be very sick just from the inflammation. Okay. There are also some carbohydrate receptors like mannose receptors. There are lectins, which are the carbohydrates that these can bind to. So, a mannose binding receptor will specifically bind the carbohydrate mannose. There's one called dectins, which is a pattern recognition receptor for fungal organisms. Um, oh, yeah, this one is actually important. DC sign. DC sign is a type that is specifically exploited by HIV to get into the cells. So worth knowing. The scavenger receptors that I think comes up actually in another chapter again. It is an N-formal peptide or metlu-Phe receptor. So these are thought of as receptors that are chemoattractants. So they actually kind of allow this sniffing effect, or it helps things move up against a gradient or towards a gradient. They mediate the uptake of oxidized lipoproteins, and they can be responsible, or when gone wrong, for recurrent aphthous ulcers. Okay, moving on to different cells of the innate immune system, I think probably the one that is the most obvious are epithelial cells. These produce a physical barrier. Of course, it's the very first line of defense all the way across the body. But it's not just a physical barrier. Epithelial cells produce mucus, they produce antimicrobials like defensins and cathelicidins, and they actually have lymphocytes that are part of the innate immune system that really have not undergone that adaptation with the DNA rearrangement. And kind of bridge between adaptive and immune, but are really considered part of the innate immune system. Right, so here's just the same thing. The epithelial cells, it's a reminder, is not just a physical barrier to infection, but it can also secrete local peptide antibiotics, defensins, cathelicidins, and it will contain some of these intraepithelial lymphocytes that have not undergone a gene rearrangement and become part of the adaptive immune system. Um, Phagocytes, like macrophages and neutrophils, part of the innate immune system that will internalize and kill microbes. They also produce cytokines and they'll repair damaged tissues. So one example, of course, is the dendritic cell, which we talked about as that very first cell that recognizes and more than actively continuing to kill the microbe or the infection, it jumps in the lymph, it goes to the lymph node, and it will kick off the adaptive immune system. There are also these lymphoid cells that come from that same common lymphoid progenitor, and they also have these same transcription factors that we've started to talk about before, like TBET, GATA3, and War gamma tau, that you should start associating with Th1 and Th2 and Th17. But in here, these are still part of the innate immune system, and they're a little bit different. They still have similar characteristics to the Th1 pathway, the Th2 pathway, and the Th17 pathway, but they they secrete a slightly different cytokine profile. But still, if it goes down this Tibet pathway and it becomes an ILC type 1, it will secrete interferon gamma and be a primary defense against viruses. If it is GATA3, the same as Th2, but instead if it becomes an ILC2, it is going to secrete IL-5 and IL-13 and be important in the defense against helminthic parasites and allergic inflammation. And then Rho gamma tau, ILC3, still very important for our IL-17 and IL-22, which is it has its effector function at the intestinal barrier. Okay, NK cells are the next cell in the innate immune system. They will perform their killing function without the need for clonal expansion and differentiation, unlike other lymphocytes. They express CD16 and 56 without expressing CD3. This is how we recognize them when we do flow cytometry. And they are regulated by a balance of both activating and inhibitory receptors. And depending on how many of each receptors is activated, it will tell the NK cell to either kill or not. So they recognize MHC class 1, and their survival is dependent on IL-15. And this is very important because if you have a form of SCID that's shutting down IL-15, then it's an NK negative form of SCID. Okay, so the inhibitory receptors on NK cells are called ITIMs. We've heard this before. It's no different for NK cells. It's in the cytoplasmic part of the tail where the ITIM sits, and it will block signaling pathways. It will recruit phosphatases, which are inhibitory. It will remove those phosphates that are activating. So examples of these inhibitory receptors that have ITIMs are KERS and CD94 and KG2A and LIRs as well. There are activating receptors on NK cells that will contain the ITAMs in that tail. They promote killing, they recruit kinases, and they cause phosphorylation. The phosphates will start these. The phosphatases remove the phosphates, and it's inhibitory. In this case, it causes phosphorylation. And there are examples of MICA A, MICA B, and CD16 are all activating receptors on NK cells. Okay, so here is just an example that shows engagement of an inhibitory receptor on the outside of a T cell, and if it is all inhibitory, then there is no cell killing. If it's all activating, then of course the signal is going to be to kill the cell. That's a sign that it is viral infected and needs to be done away with. But in reality, what's happening is there is engagement of multiple receptors, and it is kind of a battle of which is more inhibitory signals or activating signals. In this example, it shows that there's an overwhelming activating signal against the inhibitory signal, and it will detect a stressed or virally infected cell that the NK will then kill. Here are those receptors again, the inhibitory receptors up here, like CD94, ILT2 in KERS. These have ITIMs on the in the cytokine tail, in the cytoplasm tail. And then you have activating receptors like CD16, NCRs, the KERS, CD94. These all have the ITAMs on the tail, all activating. Okay, so NK cells kill primarily with two enzymes called perforin and granzyme. These are very important to know. Perforin perforates the cell, it punches a hole, it allows entry into the cell, and then the granzyme is poured into the cell, and it actually is what initiates apoptosis. So you have an NK cell, and it's encountering one of your own self cells, and it is getting a mixture of signals of inhibitory and activating. If it's viral infected and needs to be killed, then it will be overwhelming these activating signals, and it will signal for the NK cell to release perforin to allow entry into the cell, and then granzyme, which will then initiate apoptosis of that cell. So here is just an example of just that. It is an injured cell or virally infected cell, your NK cell through perforin and granzyme is going to allow that to be killed off. Okay. So, like I mentioned before, there are lymphocytes that are part of the innate immune system. They kind of straddle between innate and adaptive immunity. But these lymphocytes have a very limited repertoire, so they really haven't undergone gene rearrangement to be able to identify different microbes or antigens. So, examples of these are iNKT cells or gamma delta T cells, intraepithelial alpha beta T cells, B1 B cells, and marginal zone B cells. If you have not had the opportunity yet, you may find that a patient with SCID, when you look, is still going to have some lymphocytes that get recognized in the blood. And part of this is because of this phenomena that you can still have these lymphocytes with these limited repertoire. They are mostly sitting in the epithelium and the periphery, but they can be identified and act more as part of the innate immune system. Of course, mast cells extremely important for our specialty. They are found in the skin and mucosa. We actually don't see them all. They don't finally differentiate into mast cells until they're in the periphery, so we don't see them in the blood. But they are unique that they rapidly secrete pro-inflammatory cytokines that have already been made. They're prepackaged. They're ready to release. That does not happen almost any other time. And they will secrete these lipid mediators that then work to kill helminthic parasites. Okay, there are also some antibodies that can be produced by the innate immune system. They're called natural antibodies, again, with a very limited specificity, limited repertoire. They are produced by B cells in the innate immune system, and they're made without prior exposure to the antigen. They are unique because antibodies typically recognize proteins, but these particular natural antibodies are specific for carbohydrates and lipids. So, again, this is where we kind of fall out of that normal paradigm that we see that antibodies are made by the adaptive immune system. They secrete antibodies that are specific for proteins. This is kind of where it all falls apart here in the innate immune system. Okay, complement is considered part of the innate immune system. We've talked about this before. It can be activated through the classical, alternative, or lectin pathway, but that is not it. So, of course, we can activate it, and then it will result in the lysis of the microbes. We put in these MACs and these membrane attack complexes, and if there's many of them, it would be enough to disrupt the membrane and allow it to kill. But as in that pathway, we have all these different proteins that get activated by each other. Remember, there's part discarded. So you get C3 and it goes to C3BB and then C3BBB. Well, there's this C3A. This also acts as part of the innate immune system, and it is important for inflammation. So C3B can be is for opsonization and phagocytosis as well. Um, and C5, that C5A is not just discarded. So C3A, C5A can both um, help initiate inflammation. So just a couple more receptors. There are pentraxins. These are soluble pattern recognition receptors. There are short pentraxins like CRP and serum amyloid P that we recognize as acute phase reactants. So if you're sick, we see these go up. And there's long pentraxins like PTX3. There are also collectins and ficolins. These are another examples of soluble pattern recognition receptors. Collectins, for example, are mannose binding lectins and pulmonary surfactant proteins, SPA and SPD. And then ficolins are actually just another. Okay, so cytokines that are characteristic and important to the innate immune system, TNF, IL-1, and IL-6. We see these and hear about these all the time being talked about, especially in COVID, how much IL-6 response you have, and whether that is can be an indicator of your outcome from COVID. These all recruit leukocytes to the site of infection. Okay, and phagocytosis. So we started to talk about neutrophils and macrophages as cells being very important to the innate immune system. They are recruited to the site of infection by these cytokines that are characteristic of the innate immune system. They express receptors that then recognize the microbes so then they can ingest them into vesicles, and then it is within these vesicles that they will destroy microbes. So here is the example. Here you have something like a neutrophil, a different phagocyte. It is using these pattern recognition receptors on the outside to recognize the microbe. It's not the microbe itself, but it is the pattern on the outside of the microbe. It is recognizing, so then it will engulf it. And it will form a phagosome. The phagosome here on the left will then bind to a lysosome to get a phagolysosome. And once you have a phagolysosome, this phagocyte oxidase will generate reactive oxygen species to kill that microbe within the cell. If this cannot happen, then you have chronic granulomatous disease. So if you have an impairment of that phagocyte oxidase, also called NADPH oxidase, you have an impairment or you have a mutation in one of these components of that whole complex. So each of these, GP91, P22, 40, 47, 67, four of these have been linked to chronic granulomatous disease. 70% is all X-linked. Most chronic granulomatous disease is X-linked, and it's because of this GP91 FOX mutation. Three of these, however, have been identified in autosomal recessive forms of chronic granulomatous disease, the most common being P47 FOX, but it has been identified in 67 and 22. This slide, I would know. Here, right, it's just showing what happens. So once you get the engulfment of that microbe and you have that phagolysosome, you have your NADPH oxidase system, which is made of all these different components. This will take oxygen and it will turn it into these superoxides, these hydrogen peroxides, and those are what will actually then kill off the microbe. If that can't happen, if that respiratory burst complex cannot happen, then you start developing granulomas. You start living in an impasse where the microbe is alive in the cell is still alive, neither can kill the other off. Okay, so this is almost a bit of summary of kind of putting together what we saw. Like we learned about all these different toll-like receptors, pattern recognition receptors, all part of the innate immune system. Unlike the adaptive immune system, they're not identifying the actual microbe itself. It's just making this generic assumption it's a microbe based on a pattern it sees on the outside. And this can signal for different things. It can secrete cytokines, which will then direct adaptive immunity depending on which cytokines it secretes. It can also lead to engulfment of this microbe and generation of this reactive oxygen species to kill the microbes we just talked about. It can help generate nitric oxide, which we see, of course, in asthma. And then it will also help tissue remodeling as injury is actually, or the infection has been cleared, and it's time to actually repair the injury. Okay, so these cytokines we're talking about that are very characteristic of innate immune system, they are all endogenous pyrogens. They increase prostaglandins by the hypothalamus, TNF, IL-1, IL-6. So they will also induce hepatocytes to express these acute phase reactants like CRP, which we just learned about. And in excess, it can lead to septic shock. So here are all the different places that these cytokines can work. They're really the multifactorial. Once that innate immune system has been activated, it will of course lead to activation of different leukocytes to get to the site of infection. It will increase adhesion molecules, so you can actually get your phagocytes out of your blood vessels and increase permeability so they can get to the site of infection. They also have these systemic protective effects like generation of fever for the heat, the acute phase reactants through the liver, or increased leukocyte production through the bone marrow. We see that just by an increase in your leukocyte count if someone is sick when they come into the ER. But they can also have pathologic effects, especially if we can't calm down this enough, if our immune system is really starting to get unable to dysregulated, unable to regulate itself. But it can cause low output from the heart. It can cause thrombus in the blood vessels. We see this, of course, with age. And it can also cause insulin resistance in the skeletal muscles. Okay. The antiviral response is mostly mediated by type 1 interferon. There's both alpha and beta. It activates gene transcription and sequestration of the lymphocytes in the nodes where it can then identify or help the adaptive immune system um, to initiate its tasks. It will increase NK cell cytotoxicity and upregulates MHC type 1 for recognition by some of these cells that we talked about, like the NK cell. I'm going to skip over that. But here, I think this is the last slide, is really one of the primary concepts that the innate immune system has all these tasks. And on its own fights off infection. It's very, very important. If someone has SCID and they don't have that adapted side of their immune system, they will do fine for several months. If someone is born without neutrophils, they do poorly very, very quickly within days. So the adaptive immune system is fundamentally important, but it also sets the stage for the adaptive immune system. So whatever it sees, the signal it's getting, and then decides to share, is going to be that second signal when you get that adaptive immune system. So we always talk about how the immune system wants these checks and balances, and it won't start without a second signal. Well, the second signal is largely being generated by the innate immune system. So this is where, in our field, when we're studying inflammation and we're really trying to figure out why an initial exposure to a food, for example, results in skewing towards Th2 instead of toward more of a regulatory profile, we worry about this cytokine milieu that has already been started by the immune system. One of the hypotheses is that we get kind of this leaky gut syndrome from a dysbiosis. And once we get that leaky gut, we can get something called pathobionts, which are bacteria that are normal and healthy and commensal if they are in our gut. But once they go through a leaky gut and actually get into our immune system, they can maybe interact then with the innate immune system and start inflammation. And that inflammation then causes some of the skewing. But of course, as you both know, that is um, what is under investigation now to really understand these very first steps that lead people to be atopic. Okay.